Well, so let me done. Okay. So Matheus Januario Lopez de Sousa is currently a PhD candidate at the University of Michigan in the Department of Ecology and Evolutionary Biology. Matheus began his academic career at the University of Sao Paulo in Brazil, where he received his BS in the evolutionary dynamics of mammals, first studying the evolution of rudiments, so like the grazing cows and gazelles and giraffes and bison and all that. Um, and this was in the laboratory of Dr. Tiago Quintal. And Mateus continued on in Dr. Quintal's lab for his master's degree, researching the lineage, duration, and macroevolutionary dynamics of the rudiments. Then in 2021, Mateus then joined the lab of Daniel Roboski at the University of Michigan, researching the structure of vertebrate diversity through ecological and comparative methods. And his work has been published in both the SSE's own evolution journal, as well as Methods in Ecology and Evolution. And Mateus is a regular attendee of SSE's meeting each year. I think this will be, if he goes this year, it'll be the fourth, maybe, including when it was in Brazil. Uh, so with that, I'll let Mateus give his talk today. Can we bridge micro and macroevolution insights from vertebrate comparative demography?
Thank you very much for the introduction. Thank you all for being here. I saw some friends' names in the chat. I'm really happy to see you all and colleagues too. Thank you guys very much for coming. So I put this weird title here with a question, then a longer title at the end, but I will promise to explain myself. Today, oops, yeah, we're going to talk about first about a background of everything that like unites everything. Then we'll discuss very briefly the semantics of what I mean when I say the micro and macro evolution can, should, or could or cannot be bridged. Then we talk about demographic controls of speciation rates. Then we'll finally introduce what I mean when I say comparative demography, which is in the title. And only then we reach the bulk of the talk here, which is first about sexual selection and speciation, and then about metapopulation dynamics and speciation rate. And then if I hope to convincingly get to an answer to the question that I put in the first slide here. So let's start here. Put your seatbelt because I will speak fast now. So when we look at biodiversity, we can clearly see that it is structured in ways that provoke our, or at least mine, curiosity. So for instance, if you go to the phylogeny of fishes and you look at the oldest node within, it will separate this clade here, Cladistia, Bichirs, and the other reefing fishes like Cynopteri. And if we count how many species we have in each of those two lineages here that had them at the same time to diversify, we'll see that this one has 14 species and Actinopteri has more than 30,000 species. If we open up the Actinopteri clade, what we will find again is that we see another large difference between species number of sister taxa. And if we open Neopterigi again, we see the same pattern once more. I will keep those three fishes here in the next slide as a reference. But my point is that this doesn't happen only at the base of the reefing fish phylogeny. It happens in other clades too, like Elopomorphs and Osteoglossomorphs. It happens later on in the phylogeny here. For instance, when you compare percomorphs with their sister groups. And it happens in tetrapods too, when we compare tetrapodomorph and gymnopomorphs. So what is the point of showing this here? My point is that species are unevenly distributed in clades, and sometimes sister clades vary by even 1,000-fold distance. This is pervasive. This is in many scales. When you look at large trees, I would guess that even like every tree with more than 300 species will have some sort of tree imbalance. This pattern that we're talking about, where sister clades vary a lot in terms of the number of species they contain. But it's not only the number of species that varies between sister clades. Speciation rate itself, which I'm showing here, are color-coded branches, the warm ones being the faster speciators, and the blue ones being these lower speciators. This also has a certain phylogenetic structure. with sister clades varying a lot in terms of how fast they speciate. And of course, this is not, as I kind of imply now, this is by no means a fish-specific pattern. This is, for instance, the molecular phylogeny of birds. And again, you see those blue clades, slow speciators, alternating with clades that are much faster speciators. And the question that drives me, and will kind of be the spinal cord of this talk here, is what is creating this structure? Well, the evolutionary theory, it explains how this happened, right? Since at least when Darwin put together many different pieces of evidence, we have a, broadly speaking, general theory of evolution, and which this theory tells us, or the evidence explained by this theory tells us, is that just divergence between genes and extinction that will erase some of the temporal continuity between lineages is the thing that will create the apparent discontinuities that we observe among higher taxa. This is what's described. You can read that in the original Origin of Species, but also like there are extra literature that makes this even more explicitly and discusses this in more contemporary language too. But it's not only in the origin, right? Like Meyer, for instance, put together those 5 different major contributions that Darwin had to evolutionary theory, or the evolutionary theory has to the biological sciences. Those are common ancestry, evolutionary change, natural selection, gradualism, and speciation. And basically what Meyer is saying that this accounts for a large set of phenomena that we observe in biodiversity worldwide. If we want to be very precise, we can add these 3 other fundamental evolutionary processes here: mutation, migration, and drift. But basically, if you mix all these ideas, you basically can explain how diversity evolves. So what people mean when they write papers about bridging micro and macroevolution, right? Those papers come out all the time. This one is from last year, but there are many others. So what usually we mean when we raise this type of investigation or scientific question? Usually we or people will write micro and macro evolutions of the economy, but as I said to you, those things are already bridged conceptually. We know how populations become clades. What I see when I read this literature, I see more like the economy between mechanism motivated explanations for biodiversity. So we're talking about the rapid evolution of reproductive isolation or of phenotypic differences, or sometimes if you are more quantitative genetics oriented, we're discussing the geometry and the orientation of phenotypic or genotypic variation. And those are contrasted with pattern motivated explanations for biodiversity worldwide. Things like biogeography, trends of more than a million year, in other million year time scales, large phenotypic gaps, the evolution of new phenotypes itself, and of course the variation diversification rates, which is my question here, the one that I have enough time to address in the PhD research. So this is what we'll be talking about today. And what we want here, when I talk about bridging in my in the specific way that I am addressing this question, is that I'm looking for some microevolutionary quantity, something that we can measure preferentially in populations that will tell us what makes what we will at least correlate with speciation rate. And because those rates are phylogenetically structured, they vary among clades, as I showed you. This will ultimately explain why some clades speciate faster and why some clades speciate slowly. This is what we are looking for. And when we talk about connecting population properties or demographical properties, the first framework that I really wanted to address in my PhD students was first conceptualized by Almond in '92, Warren Almond in '92, but of course you can like trace many of the ideas down to other references, for instance, Mary's ideas. But the basically point is that we know that like the formation of isolated populations will ultimately lead to a quote completed speciation, which is basically thinking like in the splitting of 2 lineages that will later be recognized as separate species by integrative taxonomic view. we know that this thing is not absolutely instantaneous, right? This takes time, which means that we can use tools from dynamical systems to study the conversions between those types of units here and how they happen. Particularly what we can talk about. We can talk about the evolution of reproductive isolation, how fast it happened between populations. We can talk about how fast those populations acquire differences between them. We can talk about genetic difference. We can talk about ecological differences that will allow them to coexist, or genetic distance that will allow them to accumulate incompatibilities in their genome, and we'll seal the reproductive isolation later on. Of course, those two things here, they can interact among each other, and they can interact with the isolation of populations itself. But the most important thing is that those isolated forms, which are evolving those differences here in those in reproductive isolation, they have to persist for enough time for they to actually accumulate all this difference and be recognized as different species. What is the advantage of adopting this framework here, right? This basically allows us to dissect the factors that can influence and set and can be the rate limiting step of the speciation process as we measure in molecular phylogenies. This also allows us to more directly incorporate ecology, and I will address this in some minutes. And of course, this framework fits very naturally into the statistical tools that we have already available to us. So what do I mean when I say that we can't put ecology and mix ecology with this framework here? Darwin was, since very early in his book, hypothesizing that ecology is a mediator of this gap between micro and macroevolution. He didn't use these words, of course, but this is how we would translate to our language what he was talking about. Specifically, he was talking about competition between demes or those isolated forms. Of course, the word demes, right? But anyways, he was talking about the competition between those populations. So what I want to use is to connect the properties of populations and of clades in a way that is meaningful to both macroevolution and ecology. And we'll do this in a comparative framework. So this is what I'm calling this idea comparative demography. There's nothing new. I didn't invent this. I just put this word here to communicate more fast. But this is basically the point here. And how can we actually do this, right? Now I got to the bulk of the presentation here. First, I addressed this through comparing sexual selection among clades, which was defined by Darwin again as the struggle of one sex in accessing the other. And why mix sexual selection and ecology, right? Well, if you go to Africa and you start collecting data on lakes that harbor cichlids, what you will find is that if you collect some information on the ecological theory in which those lineages are coexisting, and you also collect information on how sexual selection can or cannot happen in that lake, you can very reasonably predict which lakes will foster radiations of cichlid fishes here. So this sounds very promising, right? Ecology plus sexual selection allows us to predict speciation. And why would that be the case? Well, sexual selection can influence mate choice, the evolution of mate choice, and thus the evolution of reproductive isolation. We have a big set of literature that has many mechanisms that would connect those two things, variation in sexual selection intensity and speciation rate. But all else being equal hypothesis here would be that the more intense sexual selection is, the faster that clade would speciate, or that the lineage would speciate. And the reasoning here is that very weak sexual selection can very easily be reversed or even stalled through time, while more intense selection will be a stronger selection pressure for the divergence. However, when we look at the comparative literature, we see a division in between results that find such a connection between sexual selection and speciation, and another set of literature that does not find evidence for this connection here. And a major point here is that usually we evaluate sexual selection, we compare sexual selection through the use of proxy traits. More commonly, we use sexual dichromatism in birds as a operationalization of sexual selection intensity, or sometimes people use to sexual size dimorphism, or other clades, depending on the taxon. But the question that I was trying to address when I was looking at this issue is that can we measure sexual selection more directly? And well, this is what I did together with my good friend here, Dr. Renato Macedo Rego, which is now a postdoc in the University of Viçosa in Brazil. We just submitted this manuscript called The Evolutionary Ability of Sexual Selection and Its Implications for Speciation in Macroevolution. And in that manuscript, we measure sexual selection to some indices that we call opportunity for sexual selection indices. How those indices work. Imagine that we have this population here with 5 individuals of each sex, and we will count some measurement of success. For instance, mating. We have this situation over here, and then we just count how many individuals are in this population and which is the success of all of them. So this first individual here mated with 4 other individuals. This individual here mated with 1 other individual, and these 3 individuals here have not mated with anyone. If we take the variance of this vector here and divide by the squared mean, we will have a value here, which basically tells us how skewed the distribution in mating success is. Let's compare this with a different species in which all individuals have mated with exactly 1 other individual. There is no variation in mating success here, and so our index is 0. So, of course, there are many ways to evaluate what success means, right? We can count matings, we can count fertilization events, and we can count the offspring number, which is the actual conversion of reproduction into fitness, right? But if we measure success through these different operationalizations, we will end up mixing different amounts of natural selection. For instance, offspring number will have natural selection related to viability selection, for instance. And while mating has much less natural selection components, it doesn't incorporate sexual selection that happens after mating, after the mating moment. So we want to capture many different measurements of success because we don't know exactly which one can or cannot capture more sexual selection that is meaningful to speciation. So we are trying a lot of different measurements. But in any way that we do this, a big advantage of doing so using those indices is that this increases enormously the amount of clades that we can use in our comparisons. Not all clades have dewlaps as iguanian lizards, or not every clades have feathers like birds, but all clades can technically have, if we are able logistically to measure, they all have a reproduction skewness, and we can measure this. And this is actually what me and Renato and Dan did, which we assembled a phylogeny of those 5 orders of vertebrates here by basically grafting the molecular phylogeny of every order individually into this backbone that I made based on the fossil record. And we also searched on the literature following meta-analytical procedures we measured the opportunity for selection for all these species when we could, and through many different indices, which are those 5 columns here, and we measure for both sexes. The advantage of doing so too is that this is checkable, traceable, and verifiable thus. And I showed you the variance-based indices, but we also collected Bateman gradients, but I will not have time to talk about them. We can address those later in the question section if anyone wants. We also collected speciation rates from the primary literature from this work here by Cooney and Thomas. We also used the sexual dichromatism estimates from this work here led by Jessica Armenta, and I took sexual size dimorphism from the primary literature for both sexes, or personalizing as weight, body length, or SVL, depending on the taxon. And again, our prediction here is there would be a positive correlation between intensity and a proxy trait or speciation rate. So what happens when we actually see the points here? Here I'm showing the log transformed selection intensity in the x-axis, and here we show color dichromatism in the bird clade as a linear measurement. Males in being panel, females in the other, and when we add the points, we see no significant results here. And we are incorporating a fair amount of dichromatism here. If you live in a place close to Ann Arbor, you will recognize those two birds here in our data set, the red-winged blackbirds, which this is the male, this is the female, and the house wren, which are pretty monomorphic, if you ask me. And they have actually reasonably similar intensities of sexual selection, which was pretty surprising to me. But this is just one proxy, right? What about the other? Here again, I'm showing size dimorphism in the y-axis. And all this gray area here, it means that the males are larger than the females, okay? This panel will show the male selection intensity, and this panel will show the female selection intensity, and I will color code the points by the vertebrate order. When we add the points, again, we see no significant result here. This looks like someone just thrown some confetti in the scatter plots here. So, long story short is that there is no correlation between proxies and sexual selection intensity. And when I look at this result, I was very excited, right? Because, okay, that's cool. That means that maybe our proxies are noisy. And thus, the question is that will those things correlate with speciation rate? This is what I will show here. Selection intensity again in the x-axis, speciation rate log transformed in the y-axis. And when we add the points, we see no correlation for birds, no correlation for amphibians, and actually no correlation with any other group of vertebrates. This again looks like a confetti, the end of a part that throw confetti everywhere. So we see no correlation between speciation rates and sexual selection intensity. So I was, exploring this result further, and then I decided to measure the phylogenetic signal of those indices for the opportunity of sexual selection. And what is phylogenetic signal, right? This is basically a measurement of autocorrelation between closely related branches in a phylogeny. And we know that many traits have that. For instance, more commonly, we measure body size, and we know that it has phylogenetic signal. We can measure other types of traits, for instance, those related to physiology, morphology, and ecology, or behavior. Blomberg and collaborators did exactly that in 2003. And what they found is that behavioral traits, those here, are more labile. They have a less phylogenetic signal than other types of traits. And we can recover this result in our phylogeny here. This blue line that I added for a reference is the phylogenetic signal of the body weight measurements that I took for the SSD analysis, for the dimorphism analysis. So we can recover this phylogenetic signal in our phylogeny. And the question is, what is the phylogenetic signal of sexual selection? When we add the points here, what we find is that the indices for the opportunity of sexual selection and the Bateman gradients for both females and males have much less phylogenetic signal than those types of traits here. So maybe maybe sexual selection has a very unique phylogenetic signal. This was very, very interesting, but I didn't stop it there. Then I decided to look at the interspecific variance in those indices for sexual selection. So we have some measurement, more than one measurement for some species. And what I did is that I will show them as a distribution here. But instead of showing the points, which would be different populations, to make the plot more simpler for the presentation here, I'm just showing the range and the species average. And then we'll compare this with this box plot here at the top, which is the whole distribution of all vertebrates for that index and sex. And what we see when we make this plot here is that even this populations of the same species, those horizontal lines, overlap greatly with the whole vertebrate variation in intensity of sexual selection. This is very, very surprising. I was not expecting to find this. And this happens for many indices, and this happens basically to all indices, actually. So what we can conclude here on this first half of the talk, our indices for the opportunity of sexual selection do not explain the major contrasting speciation rates that we observe among clades. Sexual selection can still be important to the evolution of reproductive isolation. But if you remember that framework that I showed you guys earlier, Other things are also important to get to make or to for a full-fledged species to appear. And it's not only reproductive isolation that will necessarily convert to speciation as we measure in molecular phylogenies here. Moreover, what we also find is that sexual selection is highly bioevolutionarily and it's very variable in many scales. It's variable among clades, even closely related clades. It's variable in intraspecific levels too. And of course, those indices are not perfect. They can vary by sex. They can vary depending to some sampling choices that, and we try to control for some of them in our analysis. They vary in our ability to recover the relative rank of selection intensity depending on the reproductive system that certain species have. This was well explored by Jonathan Henschel and collaborators here. But still, they can capture wide differences in selection intensity. So can we have a biological explanation for these findings? Well, I would guess that just mapping selection to traits is very complex. It's less straightforward than one can initially think. And the good news in certain ways that the ecological context that a certain lineage has can be more predictive of sexual selection than phylogeny. Which type of context I'm talking about? I'm talking about genetic architecture, I'm talking about social system, I'm talking about ecological theater, and all these details may matter a lot to set the intensity of sexual selection. And if those things matter that much, maybe that explains why sexual selection intensity does not convert monotonically to speciation rate. Okay, that's really cool if you like sexual selection, but now I have to address very quickly metapopulation persistence here. So we have those other controls here. And when we look at the literature, what we find is that persistence is the less mentioned control of speciation rates. So I decided to address this as an additional chapter of my PhD thesis, in which I ask if persistence is a major speciation rate control. I did this work together with Professor Malin Pinsky from UC Santa Cruz. And for we to answer this question properly, we have to find a way to measure persistence in a way that is meaningful and comparable across species, that is possible to be estimated, that has phylogenetic signal. We learned in the sexual selection part that this is important for speciation. And also, of course, persistence as measured in metapopulations have to correlate with speciation rates. And what is the measurements of persistence that we are addressing here? We are using concepts from metapopulation ecology, particularly Levin's-like metapopulations. And to understand what those metrics mean, we have to do the mental experiment of extirpating many species from many patches in this geographical lattice here. And we see what happens afterwards, how the metapopulation recover after the disturbance happens here. Well, metapopulation theory will predict that slowly this metapopulation will recover this odd equilibrium number of patches occupied here, and which we can measure based on rates. And if you have a very large grid, basically what we're measuring here is how common that species is in space, right? We can also measure how fast this recovery happens, and we can, which also depends on the rates, and we can use that as a measurement of recoverability of populations. And then the good news is that we can compare different species by how fast they recover this spatial turnover here, this spatial average occupancy, let's put it this way. This black species here very quickly from T0 to T1 recovers its average, its at equilibrium spatial occupancy here. While this blue population here does the same thing, but much more slowly here. We can compare not only just how fast populations reach this equilibrium, but also the equilibrium itself. Here again, I'm showing in gray this population that very slowly recovers its small number of patches of equilibrium, and compare that with this red population here that very fastly recovers its occupancy at equilibrium here. Moreover, what we can do is that we can take those two measurements of persistence and we can align them to make a persistence space, which you can also think that is just like a bivariate plot of those two properties here, the occupancy at equilibrium and the turnover rate. And if these persistence metrics are meaningful to the way in which a species persist, similar species in ecological terms, or maybe in phylogenetic, closely related species will cluster in this persistence space here. Of course, I've showed this just the archetypes here of the extremes, but we should expect that this space is a continuum space here. And what are the advantages of doing this, right? This allows us to make a more comparable measurement of persistence. This maps into properties related to persistence. So this will not measure which species persist more. This will measure which species persist differently. But still, it will allow us to directly base our estimations or measurements of persistence into direct observations of extirpation events that we observe in ecological decadal timescales. And we did it so. We used this data set put together by Ryan Batt and collaborators. And this data set incorporates 9 different regions throughout the US coast. Each of these regions was sampled by a different range of years, between 10 and 31 years. They also vary in number of sampling points, those cells. Some have 24, some have more than 100. And they also vary in terms of number of ray-finned fishes acting up the regions that those regions have. And the way this data was obtained was through bottom trawling, which is basically dragging a very large net through the bottom of the ocean. And by that, you basically take a snapshot of the fish community in that year in that particular place. So I hope to have convinced you that this is a meaningful and comparable measurement of persistence across distantly related lineages. And the question here is if these have phylogenetic. The way we would see phylogenetic signal in this type of estimate is if we see that some clades will cluster in particular regions of this persistence space. So for instance, this clade here, which could be a family, for instance, only occupies this quadrant here. This clade here would only occupy this quadrant. This clade would occupy this quadrant, and so on. When we actually look at the data, this is exactly what we see. We see some clades clustering in this blue space here, for instance. We see this happening in multiple clades. Some other clades will cluster in the red part of the persistence space here, and we actually can formally test the phylogenetic signal, comparing that with Brownian motion, and the p-values are significant when we do that. Moreover, we can use some combinations of species to measure the amount of variation that is explained by different factors. For instance, biogeographic region here, this black bar, and which will capture biological effects. Biogeographic regions vary in terms of abiotic components, biotic factors too. But because our data set kind of like mixes the sampling and the geography, we have this amount of variation explained by biogeographic region that we cannot be sure if it's only about biogeography or if it's also affected by possibly sampling. Those green bars here are just sampling. We know that for sure. And we can also compare how much of the variation in our data is explained by phylogeny, which are those purple bars here. And when you put together all the variation explained by taxonomic ranks or the interaction between biogeographic region and family, indeed you have more or less the same amount of variation explained by phylogeny and by biogeography. And together they explain much more of our estimates than just sampling. And this is the residual variation. The variation is not explained by any of these factors here. We have different ways of measuring the variation that is explained by phylogeny in terms of persistence. This is basically an R square here. We can look at how tall this is, and this you can picture this as how much variance in the data is predictable just if you know the phylogenetic relationships of the species in a certain data set. All those different box plots will be different sets of species that I could test for additional factors here. But the point of showing this part of this line here is that phylogeny allows us to predict quite well the persistence space, which is just a different visualization of the phylogenetic signal that persistence has at the metapopulation level. However, if we take the additional variance, the one that is not explained by phylogeny, and we partition this and the amount of this residual variation among many different types of traits, we're talking about ecological traits, habitat-related traits, life history traits, morphology, physiology, and reproductive traits. Those traits actually does not increase very much our ability to predict persistence, but phylogeny does so. Very curious. And I'm still wrapping my head exactly what this means, because that was my expectation, right? The traits will predict persistence, but this is not what we find. But okay, this is not what motivated the question, right? It was a speciation. But what about speciation? Does it correlate with persistence at the metapopulation level? Well, we have a lot of phylogenetic independent variation in speciation rates in this data set here. So we have a reasonable power to detect a correlation. And the prediction here, given that we have a bivariate persistence space, is that if persistence correlated with speciation, the species which speciate faster would be clustered in a specific region of this persistence space, right? For instance, like it's here, not necessarily in this quadrant, but in a specific quadrant. So does this happen? I will show here the two axes of persistence and the metapopulation rates here, and I will add the points and I will color the points. Each point will be a species, and I will color the point based on the speciation rate of that species. When we do that, we find no significant relationship between metapopulation dynamics and speciation rate. Moreover, if you start cutting different parts of this data set here, you see that both the slower and the faster speciators occur everywhere in this space here. So indeed, there is no sign that metapopulation level persistence correlates with speciation rates. What can we conclude here? Persistence, using the way we measure, is not related to speciation rates, even though it's weakly correlated with some sets of traits. I showed in that slide previously that the R-squares are small, but many of those relationships were statistically significant still. In my opinion, this just highlights that metapopulation persistence has phylogenetic signal, and it varies according to clades and also biogeography. So we have to explore this further and see if we want to understand this type of persistence that clades and species have. What we can conclude when we think broadly about the two things that I showed, sexual selection and persistence. Well, we now know that some demographic properties have phylogenetic signal, for instance, persistence, and some other demographic properties, for instance, sexual selection intensity, will not have phylogenetic signal. This is pretty interesting, if you ask me. I was surprised by both of these results in many ways. And the good news is that we can explore this further. We can explain and be more explicit about how much biogeography affect our estimates, how much spatial gradients and clines affect that. And we can do this in shallower phylogenetic scales, and we can design dedicated sampling designs that we can enrich through natural history and in ecological context to explore this further down and see what else we can learn about demography by comparing different distantly related lineages here. What we know for sure is those measurements do not correlate with speciation, which is aligned with other results in the literature here. For instance, some investigators have measured the rate of evolution of reproductive isolation. They did so by direct estimates and by indirect estimates in birds. And what they found is that there is no correlation between those proxies for the evolution of reproductive isolation and the speciation rate. Other investigators did the same thing with the differentiation between populations, which is here in the x-axis. And again, this does not allow us to predict speciation rates in squamates in many different taxonomical resolutions here. So I wonder what this means for this question about using microevolution to predict macroevolution. And this is a very big if that I will make now. I don't have a crystal ball. I would love to tomorrow read a new paper that will find the proxy that will allow us to predict macroevolution. But here I will talk about what happens if we keep finding this type of result, that we cannot use anything that populations have to predict what their clades are doing in deeper time scales, in billions of years. What happens? What should we do? How should we think? Well, this will mean that demography cannot completely connect what we observe at decades with what we observe at million year time scales. And this, in my opinion, makes us shift our focus back to more typical macroevolutionary occurrences here. What am I talking about? I'm talking about historical contingencies here, highlighted in yellow in this table from Jablonski. I'm talking about developments, which are those green blocks here. I'm talking about the hierarchical effects. that biodiversity may have highlighted here in red, but also there are things that we not totally fully understand. There still remain as open empirical mysteries. And a reasonable thing to imagine is that maybe our data is too sparse or incomplete to study these things. I hope this is not the case, but this is a real possibility. And we have to start thinking hard on how we can learn more about those topics that I put here in blue. Of course, there are additional results that are not necessarily in this table here, but I think they are particularly important. We are talking about the effects of biogeography. We're talking about things like macroevolutionary singularities or evolutionary perfect storms that Jablonski himself called in a different contribution, or even the ability to respond to natural selection, the ability to evolve itself. We don't know yet, but I think it will be very fun to try to find those things out. This is what I have for today, but before I finish here, I want to thank everyone that helped me do my research, everyone in my lab, my committee members, Nate Sanders, Gideon Bradbirds, and Johannes Fofopoulos, everyone from the University of Michigan that supports my research in many different ways, my friends and colleagues that discuss those results with me, and especially all the thousands of researchers, students, and enthusiasts that contributed and compiled and organized all the many databases that I used to do my research here. A big shout out to them. And this is what I have for you guys. I will be happy to take any questions that you might have. Thank you guys very much.
All right. Thank you so much, Matheus. A ton of data to digest there for the audience here. If anybody has any questions, you can unmute or type them and I can read them out. And I'll give everybody a moment to do that. I wanted to ask, so these were mostly, or this was mostly invertebrates, which is broad, but is there, are the results consistent in the literature if people are looking at invertebrates or plants?
Yeah. In terms of we not being able to predict speciation, they are consistent. But I think those other clades are even less understood, right? Like, and also they vary a lot in terms of life history. I think we have an even more incomplete view of the demography of insects and plants than we have for vertebrates. This is just a mere consequence that people, some people like to study vertebrates a lot. And so I think the unknowns are bigger when it comes to do the exacting framework to study plants and invertebrates in general. But with that said, maybe because they have more variation, it will be more rewarding, and maybe we'll be able to find things we can, because we have more variation. We can detect effects that have a smaller overall effect on speciation rate. But if we are there in terms of accumulating data, is an issue on itself. I don't know yet. I focus my research in vertebrates, but I think this is a reasonable way to approach the question too.
No, it makes a lot of sense. I'm just I agree with you. I was just thinking if there's so many species of insects, maybe you could get enough power there.
Exactly. But it will also depend on how much they vary in terms of speciation rate. We have to not forget it too.
Not just the total number, right?
Exactly.
Yeah, so I'll ask a question from the chat here. Do you think about adding social selection as a component of your research? From Renata.
Yeah, I think, well, I think that the social system of species is important even for the for we to describe well and compare well sexual selection intensity between species. That's for sure. If we're talking about different social systems, I totally agree. We're not talking necessarily about that. I probably need more details to discuss. But I think this is an important part of the natural history of these organisms. This will shape how they will interact. This will shape how their populations can or cannot address some environmental variants, for instance. And so it is potentially very important. How much important we have to measure and we have to compare. I don't know, but it's probably would be very, very interesting.
Really great talk, Matheus. I had one question that I thought it's kind of broad, but like, have you thought about interactions between processes? I was thinking in the case of your metric for sexual selection, the variance in mating success, right? I can think that like spatial structure could increase variance a lot in mating success, which is kind of related to your second part of the talk. So have you considered interactions between the processes?
Yeah. You mean genetic spatial structure or just the structure of the environment itself?
I mean the ecological part, but that could potentially drive at the genetic level, right? But I'm thinking like you can think of an idealized right fisher population will have a lot less variance in mating success than like a patchy population that exists in space.
Yes, I agree. We have very good reasons to expect that there will be such variation, and the unknown is how much of that is like so variable in time, like in decadal time scales to different reproductive cycles, that it raises any sort of like snapshot of geographic variation that we may observe. To the point, I understand this is unknown too. I hope to inspire additional measurements of this, and people can compare, for instance, two different ecological theories with the same species, for instance, and through time, and we can try to separate the effects of just like stochastic variation, for instance, between years, to more more structured spatial variation, so like a big climb in resources, for instance. We know that individuals will tend to accumulate more in places with more resources, for instance. And because there are more individuals, maybe the competition for mates will be more intense there, for instance. This is, in my opinion, likely to happen. But we have to measure and we have to compare the effects of this, let's put it this way, this stochastic wobbling in the intensity of sexual selection versus the things that are more constant through years, through ecological time scales at least. If those things hold for thousands, millions of years, is a separate issue.
Thanks.
All right, and I have one last question, I think. Like at the end of the talk, you were showing how a lot of the previous research is consistent with what you were finding in a lack of a relationship. But, and maybe I'm confused here. I could be confused here. In the beginning of the talk, you showed like a series of papers that like agreed with the relationship and didn't agree with the relationship. So there is some conflicting literature. What do you think they did to find whatever they did with the relationship?
Well, I think that the lack of correlation that we observed between the intensity of sexual selection and proxy traits has to be explored further. Like maybe opportunity changes are not capturing the sexual selection that is relevant to speciation. But this is the best direct measurement that we have to skewness in reproduction, which should be how we conceive sexual selection, right? So how can a trait correlate with speciation? Well, this could be an artifact of some sampling, particular resolution. This could be because, for instance, we know that speciation rates have phylogenetic signal, right? Maybe the traits also have phylogenetic signal, and this kind of covariation is what is driving this statistical significance here, as it happens with the traits and the persistence in metapopulations, for instance. If you look at the R squared of those finds that find a statistical significance between the traits and in speciation, usually you see that the effect size is very, very small, very, very small. In meta-analysis of this pattern, they find a very, very small effect. So I would find that suspectful. I think we have to basically open this hood furthermore and be sure of what we are measuring, what are our proxies measuring, what those things mean, especially because I was addressing two proxies that are particularly in more skeptical that capture sexual selection, like chromatism, because females can evolve to be more cryptic, and this will create dichromatism. This is natural selection. And also size dimorphism for the bulk of the theory about how size dimorphism evolves, usually hypothesize that the effects of natural selection are more intense and more important than the effects of sexual selection for size dimorphism. So there may be other proxies. For instance, social system itself, I think those will capture much better the intensity of sexual selection if mates monopolize individuals of the opposite sex or not. This will capture better, I think. So it will be a better proxy, but this is not most of the literature. It's about dichromatism, size dimorphism, and our results reject that those proxies are that meaningful to compare sexual selection of species of crows.
Got you. Yeah, because I was thinking about how hawks or birds of prey will have a larger female, but that's not necessarily a sexually selected or driven by sexual selection necessarily.
Yeah, it's actually like those two types of selection, they can mix, right? And they will mix in
Yeah.
different proportions depending on the ecological theory. So on a second thought, it's not that surprising that sexual selection is not structured among like big clades. It doesn't have a very strong phylogenetic signal because it depends on a lot of things, even things that change through decades. So under the light of that, yeah, it's not surprising. But then why we observe the other things that made us hypothesize that sexual selection is important in the first place? Big problems.
Yeah.
So as scientists, we should like problems.
Right, it's good. It's a great mystery that you're untangling.
Yeah.
And it's really, I think I speak for the whole audience when I say it's got me thinking and really great to hear. So unless there's more questions, we're just about at 1 o'clock here. So I'll let everybody go, and wanted to thank Matheus a lot for his talk and for participating, and we will see you guys here next week. And see you guys at Evolution.